Uh, I want to check if the voice is okay. Anyone can respond for that. Okay, great. This is long time. This long. So, okay. Very good to see you again today. How are you? So, anything fine with you? Anything changed from? Yes, yes, yes. So, anything changed? Something? Gain weight? Drink so much, or travel somewhere? I mean, it's a local place. Okay, so quite hard to get a response, right? So we will have the lesson today, and oh, hello. Okay. Quite not many, not all the class, but a lot of people is come already. So we can start. The one thing I want to confirm is maybe next 2 weeks we will have the midterm about a sales thing. So every year the sales examination, it kind of we have very long. I don't know very long because we have the multiple choice stuff. But in this semester midterm, it maybe cannot take part in the multiple choice format because they're kind of already the guideline from our university, so we cannot take a multiple choice because it's hard to organize the test like that. And I need to think about, no, I design ready, but it's not fixed the question yet. So maybe I can get you the kind of a writing question, and you need to apply what you learn from the class. For explain and explain for my question and answer and write out for that. So more detail I will tell you in the next week when I fix the scene and also the format of the test. So what we learn and we will test for that. And I also is kind of the scope, because I knew it, the book is too much, and you see it's any kind of chapter we have about 30 to 40 pages, so I cannot test all of them to you, and you also is quite hard to get grab all of them in your test. And I knew it, so I also minimize and also scope for some subject you need to focus for learning. Although you have to read the book and also understand all the stuff in the book, that is what I design, and also is also very good for you to understand another subject in the biology course, biotechnology. Okay, for today I continue. No, it's not continue. So last week I didn't touch this one yet. So we'll touch. Actually, I didn't share the screen yet. Okay. Okay. So in last week, what did we talk about last week? Hello, Someone talk to me again. You cannot download lecture file. I can send it to Blackboard, please. Lecture file. What is lecture file? Oh yeah. Sorry, I forgot. Totally forgot that one. I will send a bunch of the PPT file for you in the Facebook or Blackboard, so anyone can download all of the 6 or 7 lecture after today. Sorry for that. Okay. Thank you. I can minimize this one. Something I need to mention before the class. Yeah, another thing is I need to decide the assignment, because in this class I didn't send any kind of assignment for. Because of assignment take about 20 to 30% of the score. So I'm thinking about the format of assignments, not just simply write something in your home. Maybe we had some presentation, but the class is too about more than 80 people. So I don't know how we can take this presentation for all people. Okay, so we focus back to the class today. So we will talk about the DNA replication, repair, and recombination. Actually, we focus on DNA replication and repair. So I think all of you already know about the DNA replication already, right? And replication and reparation, I'm not sure, but about genetics, you learn about that, or some course about molecular biology. So you also know about DNA replication. So I just kind of get fast those things related with DNA replication. Just focus on something I know in my experience and also my search for using some knowledge about DNA replication. We can apply in biotechnology and also in the preparation of for correction of the DNA mismatch, the error in the DNA, for apply in another course in the future. Maybe you got it later, like a disease diagnosis or some kind of understanding about disease. And also that is also one part of this course. Okay, now we go to the first part, DNA replication. So the contents for today is we have 2 parts, DNA replication and DNA repair. And if we have time today, we'll move to another chapter, because this chapter is, I make the slide kind of very short, about just 26 slides already. And if we have time, we'll move to next chapter, is talking about the transcription, translation. It's also something new in you, I think. So for DNA replication, how to talk about that DNA replication? That means we have one cell. So in one cell, we have the two sets of the chromosome, and each set of chromosome we have about 3 billion base pair. 3 billion base pair here, and 2 sets we have 6 billion base pair. And you see that in one day, about cells about 1 or 2 days. So that's based on my experience in the cell culture for a long time. So if you culture some kind of the fast proliferation cells like fibroblast cells or lung cells, it get very fast. About 1 or 2 days, you kind of finish. You can have the one doubling. That means one cell becomes 2 cells, but very fast. But to make the 2 cells, the very first in the cell cycle is you need to make the new DNA for new cells, right? New DNA for new cells. So how about thinking carefully about that, how the cell can conduct that kind of work? Because the number of DNA is number of amounts of DNA is too much. Because it's tiny and very light, but the amounts of information in there is really much, right? So thinking about somebody maybe use a photocopy machine already, right? You just copy the one page. You copy it one page, and you also sometimes make a mistake in copy machine. So, you know, it's every semester I help another teacher for making the examination paper, but this is really cumbersome work. Many kind of some exams need to make the copy, and also organize the copy and prepare for the paper, prepare for the machine is really tough. And that is a number of pages and number of tests is kind of small compared with the number of the amounts of the DNA code in your genome. So that means the cells within 2 days they can do a lot of work using the molecular machinery. This is a top of work, and we can explore it today. Briefly, actually, we still have a lot of questions related to this one, related with DNA replication. And you know it, this is a general thing, not only for DNA replication. So cell activity is very important, and a lot of questions the scientists try to answer it, but they didn't have they didn't have complete the answer yet. So every day, any public, it's some kind of journal for a scientific journal, they publish a new finding for finding a new mechanism in the cell activity. So they can go to the RNA degradation or RNA synthesis or DNA synthesis or some kind of regulation. Anything is important, and that's very big finding because of even though we have know a lot of things about the DNA, about the cells, but still many questions still remain. And the second part is the DNA repair. Is also main part today. So you see that very easy to get error when we copy DNA, right? But actually we have the error always. But in the cells, in the higher organized like us, like human cells, we kind of have a lot of kind of the mechanism, a lot of the system, a lot of protein joined to help us to prepare the wrong copy in the cells. So we go deep into that. Okay. Another thing I forgot to tell. Compared with the bacteria kind of along with some slide here, we also mentioned to the bacteria compared with the bacteria and human cells. Because the bacteria genome kind of small, about some megabase only, some million base only, and very simple in there. So you see in the E. coli, we have just about 19 or 20 minutes for doubling for the one cell to become two cells. So that a human, we need more than that. So first is we talk about the DNA replication. So in the unicellular like bacteria, it's kind of simple DNA replication. But in the multiple cells organism, it's kind of like human cells. It's kind of complex, very complex, and acquire the accuracies between cell and cell to make our body, our cell system. I mean, any activity in our body is kind of smoothly, normally. So because the noise. So this slide shows the most important characteristic in the activities of the DNA replication is the base pairing. I mean, the one word, another word is complementary, double complementary, double helix. DNA structure. So remember, right? So base pairing enable for DNA replication. That is the most important characteristic of DNA. Allow it to doubling from the one to become two. So see that. So we have the DNA. We have the structure of the double helix. So we don't show the helix in here. So we had a double with a complementary between two strands. So we have the sense strand and anti-sense strand, and this variant strand. So the information from this strand, we can, when it break it, so new strand we synthesize using the old information, the information on the old strand. So the same process remain in the remain strand. So that's quite a simple. They're quite simple to remember, and also to imply how the DNA activity, DNA replication can occur. So you see that complementary is the most important characteristics of the DNA. Allow it can be duplicated. So they can break two strands, and then using the information from the only one strand to make a new strand based on that. So very simple, right? So, and then another next slide, we will show the machinery using some protein and how this system can work and how the cell can organize this. Okay. Next is we have the replication origin. So we call this and the same for also bacteria. We also call it ori for briefly call this one ori. Replication origin. So DNA synthesis begins at replication origin, and one origin we have the two replication pods like this. So this is one replication origin in this size, and then when it open, we had one pod and another pod in this side. So different with the bacteria. So the bacteria we have in the bacteria we have only one origin. In the one plasmid, we also have one origin in the one plasmid. If you are kind of follow some technology related with molecular technology related with you have to more manipulate on the plasmid. I do it a lot with the plasmid, and sometimes I also have some idea. What if I put two replication origin into my plasmid? For example, the plasmid very small. Plasmid about just 10 or less than that, 10 or 5 kb, 1,000 base pair, and it have one origin only. So before I also had I want to produce a lot of plasmid for my application. I want to do sequencing, I want to do transgenic thing, and I want to make a plasmid. And I want to kind of I want my plasmid. After a short time culture, I got a lot of plasmid. I mean, the replication is to be fast and very efficiency compared with another plasmid. So my idea is put two plasmid in one, two origin into a plasmid. I didn't try yet, but that's my interesting question. But I think it's maybe not work because there's no kind of, we also have the high copy number plasmid, but no plasmid have the two origin. Maybe that's in nature. I answer myself, but I didn't confirm yet. So different with bacteria only have the one replication origin in their genome because they're very short genome. In a human, you see that this I kind of figured it may be not correct because the whole genome we had just one photocopy machine. That's maybe not correct, right? So in a human, you see that this is a diagram they wrote it based on their electron microscope. Electron microscope, we can see is quite clear about the DNA replication. And this is origin. They break it in here, and then it's two replication port. this one, they have many places in the genome. They have their replication occur in the synthesis phase in the cell cycle. So no need to confirm. We confirm by the SAM transmission microscope. But that is also logic, right? Because the speed of the polymerase, you know, polymerase, right? The enzyme is synthesis for new strain of the DNA. So the speed of the DNA polymerase can move in quite about 1.5 kb per minute. But if our cell have the one enzyme working along through the genome, that's maybe very take a year or maybe million years to so less than that, 100 years to finish one cell replication. So that is logic to think we have a many replication origin in the animal cells, in the eukaryote cells. So the question is, what makes the origin in sequence and characteristics of origin? What is different between the origin? I mean, this is also DNA coding, right? What is special about the origin? Relate with another, compare with another reason, not origin. Okay, so any answer about that? This is just open question, not open question. We know the answer.
I think the sequence of the replication origins is very rich of AT nucleotide.
Oh, right. Why is that?
Because 8 is linked with T by just 2 hydrogen bonds, so it's easier to break it compared to the G and C.
Great, you remember well for that. So 80, that's correct. The origin is very rich of the 80. So 80 had just a weak link compared with the CG. So the cell relies on that to open the replication port. Thank you. Thank you, Yung. And so this also only answer. So compare between bacteria and human. You have any question related with this one? Okay, thank you. Nice. Things you mentioned about this. Okay, we can turn back. Polymerase synthesis DNA using the parental strand as template. This one is we already know, right? So this is our template, and the new strand is need to be synthesized from the 5' to the 3', and that is very strictly of the cell. Never we have the case in any kind of species using DNA like genetic material. We don't have the strand from the 3' to the 5'. No, because of the structure of the nucleotide very specific. So we have a 5' to the 3' is a backbone, and that is the only one direction. So we have never had the another direction. This is the detail about the elongation process. We have the strand already. So another nucleotide with the A, D, C, G with the 3 phosphates. In here we have 1, 2, 3 phosphates. So enter here. So the covalent bond is formed, and then they remove out is the condensation reaction, and 2 phosphates is out. So we call this the pyrophosphate, and that is continuously to make the very long strand of the DNA based on their old information, the information based on the old strand. The one thing I want to mention in here is the application of pyrophosphate in the sequencing technology. So Now we use a lot of next generation sequencing. So some kind of the sequencing technology, like somebody learned on molecular technology class already. So we use the sequencing, and we have to put the dye, right? The dye into nucleotide, the dye into nucleotide, and we use the camera and see if that color, that dye, and when we know that is we have a synthesis done on that side. But there are another ways. Another company also developed a new way to detect the signal. You see, as any kind of the synthesis, one-step synthesis, we have the one molecules of pyrophosphate, I mean 2-phosphate in link out of the reaction. So this one is also have the electric force. So we in the machine we have the very sensitive detector, and it can detect for the signal of the one molecules of pyrophosphate. And based on this one, so the machine and the scientists can recognize that we have the one success synthesis in the DNA strand. That is also its application of the pyrophosphates in the sequencing technology. It's still using a lot right now. Very smart, right? Somebody may ask about how, based on the pyrophosphate, but the ADCG, we have anyone have the pyrophosphates released when they add into the new strand of DNA. But the company know that, so they control. In a one time, they add maybe the ATP, maybe adenine, they add into there. If the pyrophosphate, if they have a signal pyrophosphate, they know that adenine they have. And next, they add T, and without the phosphate release, they see it. They have no any kind of synthesis done. So that's why it's kind of slowly a little bit, but also it's a very smart way. We don't need to very fancy dye to the nucleotide. So that's just one example using the DNA synthesis characteristics of the DNA in the biotechnology. And based on that one, we have the very cheap price for the whole genome sequencing. Okay. And this one is maybe a lot of people is already recognized already. So I just remind you a little bit. So we have replication port is asymmetrical. So in here we have the origin open first in here, and it tends for the both direction, this side and also this side. So one is you have to assume that the 5' to 3' DNA synthesis is very strictly, we have no reverse side, reverse direction. So in this direction is we have a continuously synthesis a new strain of DNA right from the 5' to 3'. So this is a leading strain. But from first looking in this one, when I was in the undergrad, so I didn't understand well this ones also because of the. So remember this part open first, right? So open a little bit first, and this one is go a little bit in here. So the new strand, another remain strand, they don't have the leading strand because of the DNA cannot go this way, right? DNA cannot go this way, so they have to go this way, but they don't have the space to go. So they go just a little bit on this side and go a bit to this side and go to this side, and then they link those fragments together to become the continuous strain of DNA. So this is, we call it Okazaki fragment. And this strain we call it lacking strain. So remember that, right? So, and the same process also is occur in this replication talk. Okay. Another one is the DNA polymerase is self-correcting. So This one is, we have a lot of things to say about this one. DNA polymerase is self-correcting. We have the correcting in kind of when we finish, or sorry, not finished yet. When we, along with the DNA replication, we have the correcting. But when the polymerase, the enzyme for DNA synthesis, so they get along and make a new strand. But somehow they have the mismatch. That means that maybe the concentration of the one nucleotide over compared with another nucleotide, and they jump to the structure of the complex of DNA polymerase and DNA. So somehow it's the new strand, new strand have the one incorrect compared with the complementary root of the DNA, A with T and C with G. So in that is a problem, and you see that because of the not perfect binding between the two strands, because we have the kind of from here we have something is kind of a little bit higher for mismatch, and they have no link between two strands. So the DNA polymerase can detect that one, and they can quickly cut that incorrect side, base and redo the reaction, redo the elongation. So they change by the correct one to the strand. Why is that? Because of the structure of the DNA polymerase, we have two parts. And you know, DNA polymerase is one of the most well studied for over another enzyme in the world, because of a lot of function related with DNA polymerase. So DNA polymerase, we have two kind of the remember the in the chapter 3 or 4 we have the protein. So we have the protein domain, right? Protein domain. Protein domain is different part of the protein, and they can do the different work. So this is a true for the correct for DNA polymerase. We have the one is the one is a synthesis new strand, and another domain is they is a watcher, and then they correct the new the incorrect one. So they just simply cut the incorrect one and remove that. So that is the activity we call is I put the name in here. That's we call this exonuclease activity. In a biotechnology, we have a lot of things related with this one. So exonuclease activity of the DNA polymerase. So so human is we have look like we have the I don't know, human is we have what kind of activity. So some enzyme in a biotechnology, we have the activity of the exo activity from the 5' to the 3', from 5' to the 3'. And some enzyme we have activity from the 3' to 5'. Okay, so that is used a lot in a biotechnology for correction. Some scientists also do is very smart way. They just simply remove the exonuclease activity. So the accuracies of the DNA synthesis will not like the old one, the original one, but the speed of the DNA synthesis will be increased a lot, and that is happen in the E. coli. So the scientists isolate DNA polymerase from E. coli and then modify that. So that also very interesting. So I joined that, and I also synthesized some DNA polymerase myself a little bit. That also is very interesting work. Any question related with this one? So if we talk about the DNA polymerase, we have a whole day to talk. Okay. Another one is we talk about the primase, another enzyme. So you see is any kind of activity in the cells we have there, the enzyme help. So the truth is DNA polymerase cannot synthesize the new strand without the without the probe, without the primer already in this already is hybrid to the old strand. So the DNA polymerase need the primer to elongate the new strand of DNA, and that's primer synthesis by the enzyme named primase. So they just come to the old strand and synthesize RNA primer randomly. So based on this one, so the DNA pole can extend it, and but this one is not happen in RNA and RNA synthesis. So RNA synthesis we don't need to primate. So we go to the next chapter and we see that. Okay, so this is we're talking about Okazaki thing. So we have the Okazaki is going this way, right? It goes this way, Okazaki, because we have a primase is make these RNA primer in the 5' side, and we have another Okazaki goes this way. So the DNA polymerase synthesis this way, and they get in touch with the old RNA primer, and then our DNA polymerase can have the 5' to 3' exonuclease activity, and then removes RNA primer, but they not remove for the DNA part, so they remove in this side, and then the ligase in the cells can help to make the phosphodiester bond between the two nucleosides, and they link between the two fragments of Okazaki. And another enzyme is better, or two more enzymes you have to remember is one is DNA helicase. So DNA helicase. So how is the replication origin can open? So we can with the help from the DNA helicase. In the biotechnology, we don't have this one. So we have in this. Somebody is learned about the PCR already, right? PCR, we need the heat. We need heat. We need to increase the temperature in the tube to break the hydrogen bond between the two strands of DNA. But in the cells, we don't have the heat like that, right? So we have to use the enzyme helicase. So helicase will be unwind the DNA double strand from the origin expand to the both direction. There are one thing related with the DNA helicase. So right now there are a lot of methods in the biotechnology using DNA helicase because of you see a DNA PCR machine. The machine is a synthesis DNA is quite defensive, and also is they require a lot of the energy to run it, right? So some scientists and some company also develop some method to DNA replication without using the PCR machine. They don't need to use the heat, so they can use the same temperature condition for DNA replication. But without the unwind of the DNA helix structure, they cannot do the DNA synthesis. So in that case, they put DNA helicase in the tube, and then the helicase will be automatically do the job like this. So they unwind the helix form. So that method is right now is you know, is in US. There one company is Abbott. They apply DNA helicase for synthesize the DNA for detect of the COVID-19 virus without using the PCR machine. PCR machine we have about 3 hours, but in this method they have to 5 minutes. No, no, 5 or 10 minutes, maximum about 15 minutes to confirm it's negative. So that's a wonderful and very talented method. And in the room temperature, not using machine, very fancy machine. And okay, so we have come to another problem. Okay, question. Yes. Zoom out. It's better, right? Okay, good. So where it come from already? Somebody is the slide is hard to recognize in the text. You can is another way is you can use a book. Okay, the book is I just follow the book from the figure to the figure, and this is chapter 6. You just follow the book and any information in there, so you very clear, and you can zoom is anyone you like it. Okay, so with the help of unwritten help from the DNA helicase, it's very good. But we come to another problem. That's the problem is like we have the unwritten, right? But in one in here, but we have a very big tension is stress in here because of the we win here, but the tension it didn't release. They turn this side. So it's a problem because of DNA is quite a very big, and we cannot turn along another structure on the DNA, right? It's kind of messy if you win like that way. So another enzyme is a DNA topoisomerase. They help that part. So they just come into the tension one and click it, punch it a little bit, and make a break, only one strand break, and then the strand is now the release. So based on the help of this enzyme, so activity of the on the strand is kind of safe. So information is about a 20 turn. So the topoisomerase will be asked one time for one tension. And we have another problem. So we have the chromosome, right? We have a 23 chromosome in one set of the DNA genome. So and in the chromosome structure, we have the centromer and telomere. Another information it gives you is that the centromer and the telomere is really rich of their repeat repeat DNA. That means it's a very short reason of DNA and repeat 100 times in the telomere and centromer. And now we move to the telomere. So anything is like you see it, the DNA replication go very well. But how about in the telomere? Because of it's go well in a leading strand. So in the end, we can synthesize it completely, right? But the problem in the lagging strand in here, because we need to primer in here and synthesize in this side, primer this side. But the problem is we will mixing this one because the primer cannot add in here because there's no any information for a primer can be synthesized here, right? So I mean, it's by the replication this time and your next time replication, and the telomere will be shorter and shorter and shorter, right? Because of we mixing this one, and this is not stable and very easy for the exonuclease activities of some enzyme in the cell will be cut this one, and also this one will be cut sometime, right? They shorter and shorter. So how can the cell manage this one? It's also very very beautiful to solve the problem. You see that the nature will do anything for you already. So you just discover it. No need to do something modification. So this is the end of the chromosome, and this is telomere repeat sequence. So we have another enzyme is telomerase. Telomerase will be synthesis. This strand add a new strand, and this one is even longer than the original one, if the activity of the telomerase is high activity. So in the biotechnology and also in the research, we use we have several cases is using the telomerase for what? You know, is one of the most important in the DNA is the stable condition of the chromosome. So if the chromosome break, that means a lot of genes in there break, and it's not stable, because if they break, they cannot go into the cell spindle and cannot go very correct in the next cells, right? So it cannot be break. It needs to be very correct from the original one. So one of very important function of the telomer, it is we have a lot of repeat sequence here, is to protect the structure of the chromosome. You know, it's the plasmid or another like circle DNA, plasmid circle. Circle DNA is very stable for long time. You see it in the, if you have the very old DNA, old sample, old tissue, even though it's built for maybe many years already, if you can have the DNA, what you think is what kind of DNA is stable? That's a plasmid, because the circle form is very stable. Because if we need to cut the plasmid, we need the special enzyme like endonuclease. Endo is mean cut inside. But in cell we have a lot of the exonuclease. That means the cutting DNA from the outside, from the end of the DNA structure, and that is true for the human chromosome. They are not circle form; they open for cut. So if without the telomere, the DNA is the DNA will be lost, because of the exonuclease will be eat the chromosome generally from the in the cell's activity. So that I also have the one project before when I was in my PhD. So I make a cell line. So I use the enzyme. The enzyme, this is the telomerase. The enzyme I put into the cell. Actually, I transfer the plasmid coding for telomerase into the cells. And then of course, the plasmid will make the protein, right? The protein, the telomerase. And they make the cells safe and they're living forever. That means we call it immortalized cells. The cell never dead. You know, it's in the cell culture. In the cell culture. So, and if each time of the process, I mean, each time of the cell propagation, the cell will be end to the dying, because the cell is about 50 normal cells. The 50 time of the propagation, they will die, and then they express some signal of the aging, because we age. So telomerase will help that process. And then we age because of our chromosome is degrade. Our chromosome degrade. If we put the telomerase in the cell, our chromosome will save, and also whole cell will save. I don't know how about the trends of the research rate based on the telomerase. Maybe 10 years ago is very hot topic, because of the, you know, some scientists want to reverse the aging. Old people want to become younger and younger. And a lot of scientists thinking about put telomerase in the cells and can reverse the aging stuff. We can do the cell level, but I'm not sure we can do it in the whole body. Whole body is, I'm not sure about that. So if you want it, you just need to read more about that. So I just share with you very quite interesting related with telomerase. Okay, so it kind of very fast, right? Already we finished the DNA replication. It's not many new information related with what you learned already and compared with another course. Okay, now we move to the DNA prepare. Any question related with the DNA replication? So I just share with you some kind of my experience related with experiment in the DNA replication in biotechnology. Any question? My project related with grain cell line. Okay. I had a project before and it's still going in Korea. Another, yeah, we can. That is normal. That's a kind of, some company making the service, stable service. So, you know, it's also interesting related with cells. So, you know, for example, you want to do the research related with the liver cell, right? Maybe you're working in the XBV or XCV for the virus infection in the cells. Or you want to treatment the cancer cell related with the liver cell, for example. But when you isolate the liver cells and you put in you culture it, or lung cell for any cell is okay. But the cells will weekly die, because of the DNA unstable, you see that, right? And some kind of fast fibroblast cells in about 50 times of doubling, they will die. Another cell, maybe liver, maybe less than 10 times they will die, and they will die. And you cannot do the research for a long time. Maybe you just some months already, and you finish. You have to finish this. But actually, never you have to research, and several months you finish. You have to make the cell become a long life. And another is you have to make a lot of cells to do experiment, right? You need to do extract the protein, you need to characterize and see what happened in the cell, and you need to treat the drug for the cell and rescue for the disease. Or another very important thing is you have to share your result to another country, another scientist, right? And with the limit, the lifetime of the cells, you cannot do that. So that is immortalized cell line is very, very necessary for that kind of job, right? So we need the cells still keep the characteristics of liver cell, or kidney cells, or any cells. Keep that kind of thing. And another more important is the cell should have a long life, never die. So that is the need of making the immortalized cell. So we have two methods to make an immortalized cell. So first is I already explained. I need to make the transgenic cells with a telomerase. So telomerase enzyme will be making the telomerase longer and longer and make the genome kind of stable, and then the cell will be kind of safe. And another one is, you maybe heard about this one. In the nature, we have the one type of the cell is the immortal cells. What? Somebody can answer? Stem cell can have a long life, but it's to maintain the stem cell condition is kind of very expensive and very hard. Yes, cancer, right? Cancer cells, like HeLa cell is the first cancer cell isolated. So cancer cell is a nature immortal cell. It's very strong cells, cancer. And they can have living forever. If you just put them the food, they can live. But other cells is if you put the food in there, I mean, a lot of the serum or something, they also die after several weeks or several years. they will die. So not several years, sorry, several phases. So cancer cell will have the thing. So I mentioned about the second way to make the cell become the immortal cells. It make them become the cancer, making them become a cancer cell. But how to make the cell, normal cell become a cancer cell, but still keep the characteristics of the cells is kind of that's a method. We use the virus. So I also transfer. I did already this one. It's a very easy thing. I also share with you what is tough thing really this work. I do it for more than 10 years and develop more than 80 cell line. Some is selling already, so for the company. So I transfer the virus. I transfer the gene of the virus. So I contract the plasmid with the encoding for the one kind of protein, special protein of the virus, and then put into the cells. So this virus will destroy the genome, and then they will quickly make their a lot of cells die, but small fraction of the cell. I mean, some cell from the big population that maybe it's very less, to become a cancer cell, Right now, maybe you will have the characteristics. The genome now is destroyed. Maybe it break not the 23 chromosome anymore. Maybe more than that. But the thing is, the cell become a cancer very quickly. And the efficiencies of using the virus protein is more efficient compared with using the telomere. Telomere is quite weak. Last time I used both methods in the same cells, and I kind of success. But if I use a telomere, the success rate kind of low. And another one is related with this work is using the what I forgot. The most tedious work is the selection. The selection it takes time, maybe 1 or 2 years to finish that, because the cell die. So any morning I check the cells and I see that cell die a lot. But the good thing is some cells still survive, and I keep going for that thing. And after 1 or 2 years, they success. But a lot of work in that way. I recruit some master and help me, but he also developed the very last. So now is the surface kind of good. So if you have the cells and you make the cell line, you can call the cells in the cell lines when they become the immortal line. So if without the immortal line, we cannot call the cell line. So a lot of things to talk about this one. Cell line is we need to selection. That means we just select only one cell, and we select a strong cell from that population and put into another place, and whole population downstream is only originate from one cell only. Okay, and stop that one and move to what? I stop that one and move to DNA preparation. So we also quickly fast this one. This chapter is kind of short, but is very important. You know, is how important our DNA, right? So we know about replication. It's simple, like last class you have it. And how about DNA preparation? You see that DNA replication is kind of a very big process. It's many places in our genome have the DNA replication, and joining a lot of the enzyme for that process. And you have a lot of process happen, like the cut by topo isomerase, And we also, even we have the help from the exonuclear activity of the DNA polymerase domain. But you say that, what you say? DNA replication is too complex process, right? Which is complex is really easy to break, right? Complex machine is very easy to break. So, and simple machine is kind of very hard to break. You see, the structure like a cockroach is they not very developed, but they span they not changed for a million years already, 100 million years. We can see the fossil of the cockroach for several 100 million years ago. So a simple one, and they can maintain the stable for a long time. But see, DNA replication really complex, right? And anything complex, easy to break. And then actually they break always. And easy to break and easy to get an error. That means you want to copy the you want to copy the 3 billion base pair, 6 billion base pair correctly. But normally it's not correct. There's a lot of error in there. And in this part, we will see what the error and how the cell can manage that. And another concern is, I wrote in here, in the evolution scenario, so we need the mutation. Mutation is the material for selection, for nature selection. And from beginning, our planet just have some very lonely, some very simple form of the life. But right now we have a lot of things in our planet. Even though we have some distinct species, but we cannot say that after a long time. So our planet is have a lot of species because of the selection and also evolution. That is the main force of the evolution is mutation. So the mutation get from where? From the DNA replication error, right? So the error make a mutation. You see it in more detail. It's good for development, but the problem is the DNA mutation normally is not good for me, for you, for any kind of individual, for any people, any individual, cow, chicken, or something. It's not good. That's true. We never have the scenes like we have the X-Men. If we have the mutant and we have the X-Men, a lot of X-Men in the movie. No. Hello? Hello? Yeah. Yeah. We have something. Yeah, internet has problems. So anyone okay? Can hear the voice, right? Okay. I don't know why. How about sharing the situation? Oh, still sharing. Okay, but this, as you see, is DNA mutation is not good for the individual in each people is not good. But for whole species, that may be good. But unlucky is some people have mutant. But Normally, the mutation is there. So the cells need some very good, stuff to make the genome is kind of stable. Genome kind of stable in this scenario, right? Compared with another simple form of a living thing like the bacteria. The bacteria don't have some kind of complex system for DNA correction. One is they don't care, and because of they have the why they don't care. They have a kind of very short genome, right? And then the replication time kind of very short, maybe 20 minutes. Some bacteria maybe longer. For E. coli, very fast. And then they can make a lot of themselves. They can make a lot of themselves. So if something die, they can survive well. So maybe that's the reason is nature don't put the DNA repair system to the E. coli or the bacteria. Okay. But some bacteria, they have the repair system. Okay, they have it. I explain later. Not in the book. Some kind of the archaea. You remember archaea, right? Archaea and bacteria and also eukaryote, prokaryote. So archaea have the system in correction very perfectly. Okay. Now is in this book is not very focused a lot in the DNA mutation, but they show, at least they show for us some very common what kind of DNA damage in the cells. So this slide, the one section show that the DNA damage occur continuously in the cells. What's the meaning of that? You know, is we have some mutation like the sunlight. We have a UV light in the sunlight, and we have the DNA damage in there. One thing, and we may have some we drink and some alcohol, maybe some cells in our stomach also suffer, and also suffer mutation. I mean, the environment about around the cell change. Maybe the replication system is not working well, and we have the mutation. That's it. But the thing is, it's not like that. That's not when is the environment change. We have a mutation. Actually, the mutation in here they mentioned. The mutation is always happen in the cell, even is the full fact. Even the full fact the environment surrounding the cell, the DNA damage will always happen. You say, here is the two most common mechanism of DNA damaging for the DNA structure. And here is first depyrination. We have here is guanine, but adenine also have this thing. So depyrination is we lost guanine in the base, and of course the DNA is without the structure of the nucleotide is not the same as here. So you see one of the consequence of this one is we have the base pair complements will be lost, right? And another one is loss in here. Okay, another one is also is very fourthly is a deamination. It's come from the only happen in the cytosine. Cytosine here we have an amine group in here, and we remove the amine group. We have the uracil. So from T, in here is we have a cytosine. Cytosine we bind with only bind with G, right? C with G. And we turn to the uracil. Structure of uracil and cytosine is very similar for only one amine group is in here. So uracil will be bind with A, right? So now is we have very easy the DNA code will be changed. And this is consequence. So the amine will be the amine here. C will be changed to U, and here it should be with the new strand, and they put A. It should be with G, right? But now is we have the A, so that we have the one mutation already. And This is the purine A. So one strain is lost A. Actually, they have just the base loss. The sugar and the phosphate is still remain. But the enzyme come and cut this one because it's not the work anymore. So another strain is unchanged, but one chain will be have the one deletion. So you see, it's, that's why we have some point mutation and deletion in the small scale deletion here. And another is also very a harmful thing. It is under UV light. UV light. So UV light is very toxic. Very toxic. Very toxic for you. So in the lab, in some labs is have the habit is open the UVs a lot, but UV actually is not good and it's the most harmful compared with another mutation. So what is the function? What is the effect of UV light for your DNA? So they have the thymine dimer. Thymine dimer is from the one string of the DNA. From one strand of DNA in here, we have the one strand here. So thymine will be make the appear we have the T connection. So in here, thymine is the plate of the base will be glued together. It's not nature like this. You can connect with another base from another string anymore, So they connect with each other and make that the conformation of the DNA will be changed, and they will stop the replication of DNA. Sometimes it's a break the DNA structure. So it's kind of toxic. In my experience, if you want to clean, that's why it's some kind of laboratory they apply the UV a lot, because UV due to the concentration of UV, I mean the power of UV, they can destroy the DNA. You know, it's normally if you want to detect the DNA, you have to detect using some DNA synthesis, right? But if you put your sample under UV light, totally you cannot. Only 2 minutes, your DNA will be degraded all time. If you use a high power of UV, I mean it's a short wavelength UV. Long wavelength is okay, it's kind of safe. But the UV is short, maybe less than 260 nanometer. The DNA will be break completely. You cannot recover it at all. And last time I also had one experience. Like I do recombination protein production. Not a problem with my experience is because I want to make a protein and I don't want to have a DNA. So I want to remove. Somehow it's my DNA and some part of the my protein, and they bind very well with the DNA. I cannot remove that one. So I mean, my sample have the contamination, and I want to use UV light to destroy the DNA. I mean, I put the UV light on my product, and I hope the UV light can break my DNA, and my protein is safe. But actually not. Protein is destroyed also. UV is very strong and destroy a lot of the macromolecules. Okay. Another I want to mention is about forensic DNA. Forensic DNA. For example, is you want to check the DNA of the beauty body. I mean, the body maybe is a policeman want to identify this crime scene, and maybe there's some cops in the field, but the DNA after exposed in the sunlight, they will they degrade almost. But then one thing is remain, maybe the plasmid remain. Another thing mentioned with UV is what you see is the white skin people is suffer a lot of people suffer with the skin cancer, right? Especially in some region in the earth, we have the high concentration of UV in the sunlight, like Australia. Like Australia is ozone is kind of very thin, so people in here in there they suffer with kind of skin cancer a lot. But Vietnamese people they not because of our skin and pigmentation is to protect us. We don't care. Okay. Next is a mention is cell possess a lot of mechanism. Okay, question. She didn't type yet. Okay. Oh, another question. Consequence of the UV light damage in here, right? They make a thymine is kind of connect together dimer thymine. So the conformation of the DNA strand will be changed, and they stop the replication. That means it's DNA polymerase come in there and they stop because that's not nature anymore. You know, enzyme is really specific for the subject, right? When they come to and move along the strand of DNA, and they come and they found something new, and then not nature, and then they will release the DNA. So we kind of has a problem with DNA replication. That's a big problem. Yeah, they can repair. Yeah, they can repair. Okay, cell is possess a lot of the mechanism to repair DNA. So, but in here, the book also is to summarize for us what is we have in our body, our cells, we have more than 100 protein joined to the process of the DNA repair. So if you understand this one well, you will understand about the cancer perfectly, about the cancer. So, you know, we need to prepare. So we have the DNA error by spontaneously of the replication activities, and also some mutation, the chemical, food, and the lifestyle has a problem. Maybe and also the UV as another kind of factor in the environment around the cells also affect to the error in the DNA. So and you notice, even though it's the DNA is protected by the nucleus envelope, protected the nucleus envelope, but we still have the problem because of the chemical can change, and also another side product of the some reaction, chemical reaction, biochemical reaction in the cells also affect to the DNA material for the replication. So we have a lot of the DNA's error, but the DNA is very good. Not because of that is why its DNA structure is still the dominant structure for the material for the storing the genetic material. It's first is a complementary, because of if we have a problem in one strain, we have another strain is still good, right? And the enzyme can, based on the old strain, old information, old and correct information, based on this one, we can fix the incorrect one. That is one thing. And another one is another one I can explain later. You can see it. So we have a 3-step. So first step is to recognize and remove the damaged base. So we have a lot of enzymes do that, and based on the types of the damage, so the cells the cell can help you to, the protein can help you to remove that thing. And another one is step 2 is related with DNA polymerase. So DNA polymerase just come in there. You know, it's DNA not just like the bus stop, they just come in the right place. Not like the bus stop, they come to the station and move along. No. So DNA polymerase is making a lot in our cells, and it come randomly. You see that in the biotechnology also the same thing. So DNA polymerase can bind and synthesize about just 10 base, or based on the different types of the polymerase, different species, bacteria and eukaryote. So maybe just about 15 or 50 base, that DNA polymerase will release out, and another DNA polymerase will come and continues to work. That is make process is continuously work. And you see, we have a kind of step 1 is a finish. That is make a hole in there, and the DNA polymerase come and synthesize. And then same with the Okazaki synthesis. So DNA ligase will come and make the post-pol diester bond for make the DNA is connect together again. So very diverse in here, but we have the 3 very common for any kind of activity in the DNA repair. What I want to mention here. And related with the cancer, like I mentioned. So why some people is very suffer with the cancer, and some people have a very long life. One example is you have to remember is one related with the elephant. So elephant have the one special gene that's a P53. P53. P53 is one member of the repair system, and they're very powerful. They can control another gene. So human have only one copy. Actually, we have 2 copies because we have 2 sets of the DNA. So and if we had a problem related with, if we had some mutation, I mean the native mutation, or the spontaneous by the UV something, and we have the mutation, not unlucky. And the activity of the P53 in our cell will be lost. And then now is a problem happen. And DNA, our DNA is it cannot repair anymore. No, actually another enzyme will help, but P53 cannot be important. But in elephant, we have about 25 copy of the P53. That means that's have a lot. So even though they have a lot of errors, so the cell from the elephant is kind of perfectly correct. Because of if the one P53 die, and 24 remain will be correct, the error. So elephant have no cancer. Yes. Cancer is kind of interesting. And related with us is some cancer, we have some skin cancer, some kind of animal, and they have the cancer. But the cancer happen, and then the cancer cell will die on that cell. And the human is still learn the story about the animal to treat this. That's kind of the dangerous disease. So you see that if we had a mutation related with the protein, and that protein have the role in the repair system, we will have a problem, right? The accumulation of the mutation will be more and more in our body. Okay. Now this figure is just illustrate for the 3 steps. In that, with step 1, we have some mutation in mismatch in here, and we remove it by first step. And the second step is we have the DNA polymerase will become and synthesis based on the complementary rule. But the thing is, we still have the break in here, and DNA ligase will come and fix that as a condensation process make the phospholipid step 1. Okay. And this is a kind of the like the circumstance for some kind of thing. Some kind of we done a mismatch repair, so we had mutation, DNA molecules we had mutation. And I also have one slide to propose one consequence of the mutation for you. So you understand well this one, right? The question is how the cell can recognize which one is error and which one is correct and which one is incorrect string, right? So how the cell can recognize which strain is answer. The distance between the two nucleotides. The distance between two nucleotides. But how about the the distance between two nucleotides? Okay, about the confirmation of DNA strand. Yeah, another thing. So in here is we have the problem is new strain with error in here because that is the old strain, right? Old strain is correct already. Old strain is correct. But the new strain is we have an error in there. We have an error in there. That's a new strain, the error in there. So they also have the A's in here. Even though it's the A's and we have a bubble structure, right? Bubble structure, like the A cannot bind with G and they take it like this. And why they can recognize the G is the parent and the correct one, and A is a new one and incorrect. Because of now is the A maybe they have some mistake. A is a correct one and G is not correct one, and then remove this one that's a disaster, right? So they have some mechanism to recognize the new and old strand. Okay, the mechanism is. Remember last week, we have the DNA methylation, right? DNA methylation. Only old strain have the DNA methylation, and new strain don't have the DNA methylation. So based on we have another types of the mark in the DNA, like histone also. So based on the mark of the DNA, so the protein can recognize which one is a new strain and which one is a old strain, and based on that, they can fix which one need to be fixed. Okay. Clear, right? This is efficiency of the repair system. I mean, this is in case of the repair system, and like the scientists already they measure it. So in a human, in the industry, in this industrial process, so we also have the wrong thing, like airline luggage system is a one loss per 150, means where it's in the one airplane they have two luggage is lost, that means not accept. I don't know. Okay, but the DNA replication is with the repair mismatch is very perfectly. You say there are one mistake for 1 billion nucleotide copy. It's perfectly right. It's more perfectly compared with the photocopy machine. Even photocopy, maybe the ink maybe had a problem with some paste, but this is not. Without mismatch repair, so one mistake we have the 10 million nucleotides. So I mean it's 300 base pair will become the mutation. Each time the cell will become two replication. But you see is our cell have a lot of cells, 1 trillion, maybe trillions of cells. So I mean we have a lot of cell replication. I mean in our body we have a lot of mutation, right? That's a problem. That's a problem. Cancer will be accumulate a lot. But based on the mismatch repair system, we have only one copy. That means 3 copies for cell replication. Only 3 copies for cell replication. Perfectly, right? Beautiful work. And the question is where those mismatches go. 3 nucleotide will change between the sister cells from the same parent cell. So they have 3. And where they go? They make the X-Men or they make the bad protein directly to your body? No. Even though we accept the 3 base pair will be have mutate, but it not go directly into your health. What's the problem with that? So remember is you didn't know it, but something you know already. So you know is our genome is we have just 2% is exon, right? 2% is exon. Remain is an intron or something non-gene. So the thing is the possibility of those mismatch, the mutation right into your exon is not. So they go into the desert of no gene there, no house in there. So you are safe. But sometimes they unlucky go into the right, very important gene. That cell has some problem, but we still have another cell, healthy cell. And you know we have an immune system. I will come back this one. It comes to the chapter related with the cancer. The immune system. So still we have a lot of cell problem based on a DNA mutation. But immune system will be detect very well which cells it has a problem, and then they kill that cells. Okay, that is why we say our cancer. Oh. Okay, now we come to the problem. So the problem is if you use a complementary sequence to fix your error sequence, but what if we have the problem like timing dimer and DNA is a break? Maybe some UV or some kind of the nuclear weapon. So maybe some very toxic agent. Make the cell is the DNA, the chromosome will be break it, and I mean it's both strand in the complementary is in the relationship they are break. So no backup information for you. So where is how can you find the backup information for fixes? So in the figure you can see that. Good thing in the human cells we have 2 sets of DNA, right? One from dad and one from mom. If you break from that, so the set of DNA from mom can help you to fix the error from that. That's a wonderful thing. We have a 2-time backup for the cells. So, but in that sense, we have also 2 strategies for repair. The first one is quite very fast, and but fast is not good, and also thorough. So in here we have to break 2 strands. I mean, SAN and anti-SAN also break, and you cannot use a complementary, right? So and another enzyme will eat this one and make there a blunt end. So the enzyme ligase will add that one, add to ligase together. But this one is what is the consequence? This one is perfectly and very fast. But from this one, we will lose some nucleotide in here, right? We will lose some nucleotide, and we have another better way. So this we call is homologous, based on homologous between the 2 sets of DNA. Because remember, is chromosome is we occur in the cell is in pair, right? In pair. In the gamete we have a long one, but in normal cell we have it in pair. So we break this one, and the DNA will come in here. Some enzyme in here will be cut, and then synthesis a little bit, and based on the remaining one, reservoir for information. So and then the final is we don't have any loss of DNA, perfectly work. But this one need a lot of DNA to choice. And you see that if some system need a lot of enzyme, the system is easy to break again. So if you have, maybe this one, they have a lot of enzymes coming in here to help, but some enzyme will be, some mutation will come to that enzyme, and and you cannot fix for this case. And the cell is a mutant cell is die, become the cancerous cells. This is a more detail about the situation. Just have a look at that. And this is related with when a mutation. If the mutation is come to any part in your body, and then they cannot go into any part, maybe into your hand or your lung or your kidney, and if the very dangerous and a very bad mutation, and they develop the cancer in your body. But the cancer cannot go to next generation. But some cells, some mutation, they can occur in your germ-like stem cell. Germ-like stem cell, maybe it becomes a sperm or the egg, and then that mutation can move to the next generation, and then they can explore maybe the whole population downstream of you. We have that mutation. That's a new mutation. Okay, that is how the species can develop the evolution. Okay, so but sometimes the cell cannot develop. Sometimes the cell cannot fix by chain. The cell cannot fix the mutation because it is a very long process, and some mutation is still remaining today. For example, in the case of the sickle cell gene here, so only one mutation in the beta globulin gene, very important gene for oxygen transfer in your body in the circle system. So only one nucleotide change. That nucleotide make the amino acid chain, and you will have problem with the red blood cells safe. This is more easy to this is normal, and this is a disease form. And another one is like I said that some kind of protector protein for repair your DNA is a mutation. So that is effect to the cancer incidence. And here is a colon cancer incidence in women. So it's based on the age. So in the young age, no problem because the cell still is not much and they work perfectly. But when we get old, the cell is kind of a lot of mutation accumulate mutation, and by chain forceability is increase. And so some mutation is now it become a very dangerous mutation, and cancer will develop. Some people is kind of safe because they have no harmful mutation, and their lifestyle is okay. Okay, and then final one is we had a mutation, right? Mutation. That is also very important for evolution, and also the mutation make the material for selection, and that is also when have enough different between this one and this one, so they will have a speciation. That means the quantity of different is accumulate enough, so they will break the species. So in here you see different, very different between the blue whale, and the human. But some genes are very important, and the nature not allowed to that genes to have mutate. For example, the one DNA segment belong to the sex chromosome. Sex chromosome is really important, and then they not equally mutant occur compared with another chromosome. So say it's any kind of species in animal only have two sex chromosomes, and then they are kind of independent compared with other chromosome. And you see, where human is very similar in that gene. Of course, another gene is very different. And also the same group. So intron and exon. So exon is very less mutation because it's quite important to pay with for survival of the organism. But intron may be less important. So a lot of mutation come to intron, but exon is not. Okay, we finished the chapter 6 in here, and we have break a little bit, about 5 minutes and 10 minutes. and we come back for the chapter 7, maybe just a little bit, maybe more than 10 slides, and we stop today. Okay. Post your question if you have any question for the chapter. Okay, thank you. You still hear me, right? Okay, nice. Another one. Yes. Another one. Come back. So we have some rain in here. It's good in the southern part. Okay, okay. Um, so like you see that the cells is quite a very interesting stuff. Is during the study of the cells, we know a lot of things related with the history of the living thing in the. We can answer some question related with history of these the living thing in our planet, and also we have a lot of application in the technology, biotechnology, and cell also like some kind of question related with my experience, my experiment. Like you say that cell also provide for us a very good tool for doing the result and research. And when you do something related with the research, you cannot do that the experiment on human or home body of the species, especially home body. First, we need to apply for the cell first, and the knowledge about the cell we have to apply in the thing. So after midterm, we have some very interesting stuff related with many parts in the cells, and I think it's very useful for you. It's quite very different with what you learned already. From like in here, you know DNA replication, like in prepare system. I remember we also don't have in another subject. And now we move to the what also is an old term is transcription and translation from DNA to protein. From DNA to protein is we have a central dogma, right? Central dogma. From we have from DNA, we have a picture of that. How does cell read the genome? That's kind of a big question. Big question. So you see that DNA is DNA just is a coding. They don't do anything. That is just storing the information for the cell can follow to operate the activity of the cells. The DNA do nothing. But the do thing is the protein. And the remain work is we have for RNA. RNA also do a lot of things in the cells. You know, like ribosome, right? Ribosome is mostly is RNA, not protein. Protein just contribute a little bit in our ribosome, and you know is how important ribosome to our cells. And like in the very first presentation, I also mentioned about some common characteristics of the living cells. One of the very important things is they have DNA, like the genetic material and what, and the tool and the apparatus to use that information for doing the work. So what is the tool? What is the tool and the means to use that DNA information to do the work?
Chứ còn bình thường nó như này. Cái mà giờ có cu đâu mà biết. Thì em cứ thấy nó phẳng xuống chứ nó không phồng ra.
Okay, anyone is somebody is open your microphone and it interfere a little bit. So please mute your microphone a little bit. And if we had a question in another part, okay. We have several slides in the chapter 7, and I hope in this we go home with this. And next week we will study about chapter 7, chapter 7 and chapter 8, and we will review for midterm. Okay, firstly, this is a picture, very famous picture, and anyone in biology needs to be remembered and analyzed, and also report or states about this one. Statement of this one, like from the gene, from the DNA, we move to the RNA by transcription and by RNA rows, and from the RNA, the signal he will translate to the protein. And the protein do many works and almost the work in our cells. So why is a DNA is from DNA to RNA is a transcription? We call it transcription. And from RNA to the protein, we call it translation. So somebody is remember that why we call it transcription and translation, because we call it process like that. So you see it's from DNA to RNA. So the coding is not much different. So it's just slightly different in the structure of DNA and RNA. So we call it transcription. So I mean a different way to write it, different way to write the sentence for the code. That's what we call it transcription, because very slightly change the DNA and RNA. Because we have just ATCG and RNA, we have UCG. But from RNA to protein is quite a different language. From I come from this country and jump to another country to say something weird. That is, we have the not nucleotide anymore. We have to transfer using the amino acid code. So we call it too much different. We call it translation in the process of the protein synthesis from RNA coding. So in this contents of this chapter, we have a 3-part. The most important, the first part, we talk briefly about DNA to RNA, talking about cell-wise, and not touch very deeply about the genetics. And the second part, we move to another step, is RNA to the protein, RNA protein. And the final, we come to quite a very interesting question. So between the RNA, DNA, and the protein, which macromolecules is the first occur in our planet? Maybe God is not moving this molecule from another planet to our planet. I'm not sure about that, but we just ask is which macromolecule is the first in our planet? So they will hypothesize. We don't know yet because nobody in that time ever. So we just based on our evidence and suggest the hypothesis. I mean, scientists, not you, and you also will try to understand that why it's correct or not. Okay, so from DNA to back to here, it's also related with what I showed yesterday. Is you know, it's from RNA we can turn back to RNA to DNA, right? by the enzyme named reverse transcriptase. So the enzyme is synthesis from RNA to DNA. Normally, in human or animal or plant, we cannot have that kind of function because of that's weird. But some virus, they have some enzyme can do that. It's a wonderful thing. So I mean, it's wonderful about the application in about technology. So you can use it for detect of the virus in the sample. I should know that you know is which kind of virus make RNA from RNA to DNA. I'm asking about the biotechnology wise. So somebody remember what type of bacteria? XIV. So XIV or lentivirus, XIV or XIV. What kind of other virus? XIV. What kind of virus? I mean, it's a family. I mean, family virus. What's this? You know, yes, retrovirus. Thank you. Retrovirus. Retrovirus is we have they have RNA and then they need to make DNA and jump into our genome. You know, is that our genome have a lot of virus, right? A lot of virus. Talking about a retrovirus is really interesting. Any kind of species have a retrovirus. Our university, I mean, since my before university. So we had a project. I tell you this one already, right? So we had a project using the pig tissue to transplant to the human because we need a lot of tissue for the people with the cancer or some injury need to be transplanted, replace with new tissue. So we think about a pig because pig it kind of had the size of tissue and also the physiology activity very similar with humans. So some scientists from England, he hypothesized that human is a product of the mating between pig and a monkey. So that is we have one paper post that one. I don't know in BBC or some kind of like that in England. So pig is very similar with human, even though they are kind of far. Human and mouse is amazing in the genetic relationship with human and mouse is more close. And pig is another one. Pig and whale and cow and like hippo is kind of close in one group. I had one paper related to that one. So like Jung said, that is that's a bit long to retrovirus. But yesterday I saw that there was another virus, not the retrovirus. They also have the activity of the what? Reverse RNA to DNA. Another virus. This is the very fourth virus in Vietnam. You know, that's an HPV virus. HPV virus is a DNA virus, but they also have the virus life is also very complex. And one of the period of time, they also have the activity of the transfer back from to DNA from RNA. So it's very interesting. Oh, I forgot. Sorry, I didn't. Talking about pig. Sorry about a retrovirus. So pig is really pig is perfectly for human transplant. But the problem in pig, the scientists worry is they have a lot of retrovirus in their genome, and somehow those retrovirus maybe wake up and they make some harmful. You see it in my experiment. So like I transfer some virus in the cell and make the cell become a cancer cell. And the scientists also very worried about the situation. And other species also have a lot of retrovirus. Human also have some retrovirus in our DNA code. So, and sometimes they will wake. And you see a lot of cancer related with the virus, retrovirus in our cell, native cells. Next is this figure show is also same with the last figure, but they show is related with the gene. You see, the whole genome is we have a very big genome, 3 billion base pair. This number is not changed for me for a long time. So, but part of the genome is can express. So what is I'm talking about the term is what is expression? Expression is that come from a DNA, can to be RNA, or the final stage is a protein. We call it expression. Expression not only say about the gene to the protein, it's also meaning that is also only the process from the gene to the RNA. You see that gene is a unit of the genome, so they can make the RNA and making the protein. So this figure is quite very important for you because of some gene, they express a lot of RNA. Some gene is very less, and from RNA is a lot of people will accept that when RNA, so they will be expressed to the translation and will be applied to translation to the protein. That is anybody can accept that. When they have RNA, automatically we will have the protein. That's it. For example, if you want to have your publication, you have a publication. You don't actually the requirement. You say that gene A is more expression than gene B, and you just prove that in the RNA level. That's enough. You don't need to do the protein expression confirmation, because the protein work is quite very hard and not many labs. I mean, it's not many labs, even especially kind of poor country in Vietnam. So not many labs can do the protein work. It's kind of complex and for, quite not very, but quite take time and expensive. So RNA is the scientist's work is accept. So from RNA we can transfer to protein easily. So in here is why we need the RNA? Why we need RNA? Because you see that why we need RNA? Because of in the one cell, we have just 2 copies of the gene in the one cell, right? So you see it, the speed of the making of protein, if the cell needs protein. For example, in the process, we need a lot of energy, and so we need a lot of protein related with the mitochondria for the synthesis new mitochondria. We need a lot of protein. So if the gene go directly to the protein, that process will be very slow, and the cell cannot control that well. So that is the one role of the make the losses of the roles of the RNA. So when the cell needs protein, they will make a lot of RNA. Yeah, and here is a gene B is we don't need a lot, so we need less RNA. So that is how the cell can control. That we will talk in next week more clear. So now is we have a lot of new findings. That new findings is what you know is the number of the gene of the genome is about 25 to 30,000 of the gene. That means your gene is almost encoding for the protein. I mean, it's exons and coding for the protein. But now is a lot of people will not accept that number. Not accept. If coding for protein is okay, but now is a lot of many kind of gene important to the cells. This is a regulation gene. So if you count for the regulation gene, so number of gene in the cells should be more than 100,000 gene. It's a lot. So that regulation gene is not required to make a protein. They just make the RNA and finish. And RNA will join to the regulation process. And we will see is what kind of regulation. So RNA not really go to all the protein. No. That's also, I'll share it later with you in my experiment. So, and the speed of making a protein also different with the different types of the gene, different types of RNA, because some RNA have the before we have ATG, right? ATG. So the upstream of the ATG, we have the 5' untranslated reason, 5' untranslated reason. So if the 5' untranslated reason is long, I mean the speed of making the protein kind of slow. If the short, the speed of making protein kind of faster. And some kind of RNA kind of not stable, so very fast to degrade, and some RNA kind of have a long line, but cannot compare with the DNA. So here is you already know is we have a chemical structure of RNA is slightly different from DNA. So here is in the right side we have using RNA, and they're different in the two things. First, all of the base in the RNA is a ribose sugar, and the DNA is a deoxyribose. We have no oxygen in here. So this is another question. So ATP that we use to for energy of the cells is the ribo or the deoxyribo. This question I asked before. Somebody can repeat it? Cast the question. So energy we use the ATP, adenosine triphosphate. We use for energy is ribo or deoxyribo. Similar with RNA or DNA. Hello. With DNA, so it means deoxyribo, right? Deoxyribo is a main sugar using ATP. Normally we use for energy, right? Another idea? No. With ribose, okay? With ribose. This is a sugar we use ATP for energy ATP. So ribose, not the deoxyribose. Why? I also searched for this why the nature using ribose sugar for ATP for energy. So the answer is not clear yet. The answer is not clear yet. But totally ATP use ribose sugar for their structure. One thing is because of oxygen here still have the high energy a little bit and easy to join the chemical reaction. That is one hypothesis is that somebody can accept this. Okay, we go to another difference between the DNA and RNA in structure is different between the thymine and thymine in DNA. So thymine in DNA is chained by uracil in RNA. So one different and the second different is we have the ATGC in the DNA and we have the AUGC in the RNA. So uracil is normally is uracil, and also another for some ribo sugar is not stable, and that's why DNA is very stable and RNA is not stable. RNA is a half-life very short and very quickly to degrade in the cells. And when you are having experiment with the RNA and you extract DNA from the cells, and it's also very easy to degrade. Somehow it's a cell is don't like RNA, so because their effects very strong. You see, our hand have a lot of enzyme. The enzyme is the RNase. It means the enzyme cut RNA is a lot in our hand. So if you want to work in with the RNA, so you have to prepare the lab very clean out of there using the hand. You have to very good quality glove and all of the all of your equipment should be clean of the RNAs from touch. Okay, so uracil. So uracil is a replay for thymine, and they will have the hydrogen bond, weak bond with the adenine. So it's the same thing. So the structure is same with the DNA. So also by sugar phosphate backbone for the backbone of RNA. And the good thing of RNA is they don't have the link. They don't have the link, double structure, double strand structure like DNA. They have a single structure, and that is make a lot of things come with RNA, very diverse. Not only sit at one place like DNA. And you see, I want to explain one thing. So you see that the DNA and RNA just different. Only one point. That's an oxygen atom. Atom is kind of very small, but you know is the DNA polymerase enzyme or many enzymes they cannot accept they cannot accept ribonucleotide for the subject. They reject. That means very hard to force. We cannot force DNA to synthesize using the RNA. So only one do that is a retrovirus using the reverse transcriptase enzyme. Right now, there's many groups in the world. They modify the protein structure of the DNA polymerase and force DNA polymerase can have the activity like the reverse transcriptase. We can do that, but the success is very limited because they can do, but very short, less than 100 base pair, and that they cannot use a lot in biotechnology. Not now. Okay. So you see, is the structure of RNA is they cannot. They have a single strand structure. So single strand structure, that means they have a free base for the complementary binding, right? So they can bind with each cell, right? So to become the secondary or tertiary, the 3D structure of the RNA like this. So many parts, and then they based on the sequence, and they have the some native structure of RNA. And some very important native structure of RNA is the tRNA, and help us to transfer the new amino acids in the protein translation process. That's it. So can we predict the RNA structure based on sequence? And the answer is for fast is yes. If you put the sequence of RNA into a software easy to recognize for you the structure of RNA. Our lab before is try to express, make a protein of some very offensive protein. That's a Botox protein. You know Botox, right? It's a lot of famous people, actress, or even the actor. When they get old, so they use Botox to put in the muscle. And the Botox is very toxic. Botox will be make your muscle will be a little bit get inflamed, so become a younger look like. But the very toxic. So it means 1 gram of Botox can kill all the people in the our planet. Very toxic one. But that Botox is kind of very long gene. And if you put a very long plasmid and then make a very long RNA expression, because we have DNA to RNA very long. So RNA very long is that they also form very complex 3D structure like this, and the ribosome cannot work well in this case in an equalized system. Okay. So this is our process of the transcription. First process is we have a coding DNA and this is coding strand. Actually, a cell didn't use this place, this strand for synthesis RNA. They use this one, the antisense for template strand, and the RNA polymerase don't care about this coding strand. The coding strand is why it's important, because the coding strand is the code very similar with newly synthesized RNA. So actually, the cell using this strand for synthesized RNA. And RNA in single strand form will be different with DNA. To say that after synthesis, even very small fragment of RNA. So the new strand RNA is very quickly disassociate, break out of the old DNA. See, they break it. They not bind it like DNA. So you see that when the newly strand of DNA is synthesized, they will bind very tight to the DNA, but RNA is not. They quickly disassociate it. Okay. And but in the biotechnology, also I like to mention a lot of things. So in biotechnology, we can synthesize RNA. We can synthesize the DNA, can bind with the DNA. That is true. We can do that. The DNA can bind with DNA complementary, very stable. But then we have to optimize the concentration of buffer and a lot of things related with that related with that binding. That's possible to bind between DNA and RNA. It means hybrid. Okay, because they also is a complementary. Okay. This is a beautiful picture from the book. So they illustrate the very active activity in the transcript. How can they activate? Because this is for ribosome making ribosome. This is a ribosome DNA, and this ribosome RNA is making. So you see that from the first time I look at this one, I think this is the protein. And I see that this is a ribosome making a protein, but no, this is totally this long strand is DNA, and they have a two gene related with the this gene is related with the ribosome gene. You know, ribosome is too important, right? And the activity of the ribosome is very strong. I mean, it's a primer or something. So a lot of RNA polymerase come in here and generally making a series of the synthesis machine, and a lot of the molecules of the RNA synthesis continuously. Okay. So this makes a nucleolus because of we have a lot of things like this. And in nucleus, they form something visible under microscope for us to see. We have some big difference between the DNA and RNA synthesis. That means that DNA RNA synthesis very specific for ribonucleotide. They cannot accept the nucleotide. That means they also nucleotide. You know, very specific. And RNA is a no-need primer. So you remember in the DNA replication, you need a primase enzyme to make the primer randomly for the DNA polymerase can attend, right? But in RNA, we don't need it. We just need the signal sequence and receive. Signal sequence and attend it. And it has a very higher error rate because of you see that RNA is quickly disassociate out of the DNA. So they don't care about the correction and quickly making for the protein. So they have very high error rate. That error rate can make a protein. Something we can make, but something cannot make because of stop codon mark or some kind of non-function change in the amino acids. So cannot make. The application of RNA for sample right now, you know, is very hot strand related with the single cell sequencing. So meaning the scientists only take one cell only, and they know what is the problem with the cell. And problem with the cell, and they know is what problem with that body. So we have a technique is related with single cells sequencing. But single cell is we have a limit of the DNA's amount, so we cannot the sequencing DNA. No, we can, but it's not complete and it's a lot of error. But RNA gave us a very good thing. Why? Because of just 2 sets of gene in RNA and in DNA, we can make a lot of the RNA. So a lot of RNA provide a lot of material for experiment and confirm that our hypothesis. So that is the quantity of RNA compared with DNA is superior. So we can use RNA to do a lot of work. But I don't like doing the experiment with RNA because of that. They very quickly degrade. They're not stable. Okay. So this is all you all know, right? So the cell is have a various types of RNA. When I was start to my master PhD, number of type of RNA is kind of not much, like C. But after that, a lot of new type of RNA that scientists discovered. In here, we know about the messenger RNA. So we call them protein, like ribosome RNA, also famous. And microRNA is quite old, but there's a lot of information on application already, we know. And transfer RNA also very famous. And other non-coding RNAs is still a lot of scientists identify. For example, is we have a non-coding RNA. For different with the microRNA in here. So microRNA kind of small, but long-coding RNA kind of very long. And what function of this one? So you remember the X chromosome inactivation, right? We had a bar body, right? So the long-coding RNA will be produced a lot in the one gene of the X chromosome, and then they coding for the whole X chromosome and reboost another protein come to X chromosome and inactivate that is the structure of the X chromosome. That is one function, very famous function of the non-coding RNA. A new type of RNA is still going. Okay, we kind of stopped today and spread this one for next week. Okay, any question related with what we already fast? Question? You still there? Anybody? Okay, so that's quite very basic, and I'm happy to share with you what I learned and also what I did like with the RNA and experiment with the cells. Some molecular work is a lot of application if you know well about this one. Okay, last week it's an accident. I forgot to put the button of the record video, so the video last week I don't have it. So I will upload the PPT file from the chapter 1 to chapter 7, maybe the chapter 8 also for you to have a midterm. And any question related to midterm you concern. And next week is fixed, but if I have any information I can share now. Any question related to midterm or related with let's stop. We have a lecture today. Hello? Yeah, that is short but important question. Okay. You may have a question, a writing question due in the short time. Anyway, same question. Or maybe it's different question. I'm not sure. But in the time of doing the exam, you need to turn on your camera. So anybody need to be prepared for. Yes, sure. We do it. Yeah. you need in the showing face in the time, or you need to show in your head also, or also you prepare the student part very short time. I test for me not a long time, showing face, and then submit your writing for me for scoring. So don't worry, I try to make it simple. And the thing in question is, I am trying to fix the question to relate with what you are learning already. But the thing is, the question is quite different with multiple choice. So you need to remember and also understand well to explain, right place to explanation. Okay, any question else? Open book. So global thing I cannot control, right? Due to you, I'm not sure that. But the thing is, I will let you know which kind of part you need to focus on writing. Thank you. Okay, have a good weekend. So I'm happy to share with you what I learned. So hopefully you enjoy that. Yeah, I will upload a video. Good thing is I record a video, so I upload this one. Last week I forgot, so I don't have it. Okay, if you have no questions, so thank you very much for your joining me today. Thank you. What?